It's Michael Woźniak, and together with my wife Monika, we founded PURA Foundation Poland after our son Alexander was diagnosed with PURA syndrome. Today we know about 18 people have been diagnosed with PURA syndrome in Poland. This includes 16 children and 2 adults. 13 families are currently supported by our foundation and take part in our activities and research projects. Our mission is simple. We connect families, doctors and researchers. We support researchers, increase awareness of PURA syndrome and help build new international collaborations. Every new patient is important. Every family brings new knowledge. Together we can better understand PURA syndrome and help future research. Since 2022, we have worked to bring together families, doctors and scientists. We organized the first Polish PURA conference in Warsaw. Last year we presented our work at the PURA Syndrome Conference in Dallas. Today, together with our partners, we are supporting the Polish Natural History Study. Our next goal is to expand this work and include more patient and research centers across Europe. We believe that patients' organization can do more than support families. They can also help make research happen. PURA syndrome is a very rare disease, and we know all of us. No single country has enough patients to answer all the scientific questions. That is why we need to work together, especially in Europe. Today, our foundation is building partnership with researchers and clinicians in Poland, Germany and Switzerland. We believe that by sharing knowledge and working together across Europe, we can speed up research and move closer to future treatments. For rare disease, collaboration is not an option, it's a necessity. Now it's my pleasure to introduce 2 people who have played an important role in this project. First is Dr. Kacper Łukasiewicz and Hubert Kubiszewski, who will present the scientific background and the result of our natural history study. Thank you very much.
Good morning, everyone. My name is Hubert Kubiszewski, and I work with the PURA Foundation Poland, coordinating the natural history study of the PURA syndrome, which we performed in the May of on this year. How can we switch the slides? Oh. So it is a great pleasure to be here today and to have the opportunity to share the results of our study with you. Together with Dr. Michał Bielak and Dr. Kacper Łukasiewicz, we have spent over 3 months planning the study, and we would like now to present our findings today. So, every medicine available on the market has had to go through the so-called pyramid of the clinical trials. At the base of this pyramid is the knowledge of the disease's natural history, the evolution of the symptoms over time. This knowledge is this is the only way to determine whether a drug is effective, by comparing its effect against the clinical state before treatment and observing whether the patient improves or further deteriorates. Similarly, if we can identify biomolecular markers, such as specific proteins in the blood, that correlate with the disease's severity, we can assess whether a treatment is effective at the cellular level. In short, a well-characterized patient cohort can serve as a reference point for evaluating experimental treatments. Our study was based on the 4 elements. The first one is nearly 30-page questionnaire completed by parents, which was sent by an email, returned to us and then analyzed. It consists of almost 200 questions, and the surveys provided a general overview of each patient's clinical state, and they were focused on symptoms potentially related to PURA syndrome. The remaining three elements, the blood sample collection, the physical examination, and the quantitative scale assessment, were performed at the hospital by Dr. Michał Bielak. Blood was drawn first to ensure the children would not have to fast for too long. Our goal was to analyze the plasma for metabolomics, lipidomics, proteomics, and transcriptomics in order to establish molecular phenotype correlations. Finally, during the physical examination, each patient's general clinical state was assessed alongside with the quantitative evaluation of low muscle tone, which is called hypotonia. We began organizing the study in February, and in May we received the first surveys from a group of 6 parents of children with PURA syndrome. On May 12th, we performed our first physical examinations with blood draws. In total, we have completed 6 surveys and collected blood samples from 7 children of Polish PURA parents. Next year, we plan to continue the study with annual follow-ups with improved tools, including the neurodevelopmental assessment conducted by a qualified pediatric psychologist. Our questionnaire consists of 24 chapters, beginning with general information about the patient and family, followed by diagnosis, perinatal history, and whole organ system overview, motor, verbal communication, and comprehension milestones, and educational background, concluding with a medical history of surgeries and allergies. Parents can raise any questions in the comment section at the end of the survey. The survey is an original instrument. It was based on reported cases of the PURA syndrome and on similar studies conducted in China. As I mentioned previously, the clinical assessment consisted of basic physical examination with height and weight measurements, alongside with the neurological examination that included an in-depth evaluation of low muscle tone hypotonia. Because hypotonia is, as we know, the most prevalent symptom among PURA patients, we consider the quantitative assessment of its severity to be one of the most crucial parts of our natural history study. The gold standard for such a measurement would be a widely accepted, validated scale capable of objectively assessing the muscle tone, not just by asking questions to the patient, but by objectively assessing the visible effects such as postural abnormalities, range of motion in the joints, and the physical strength of the core muscles. Given the limited number of tools that fit our criteria, we initially approached the assessment of hypotonia using a modified version of the Hammersmith Infant Neurological Examination, HINE for short. However, the results were unreliable due to the age of our patients, which was over the 6-year-old, and the scale was developed for the younger patients. So we would like to suggest other scale, which is called HFMSE. This is the Hammersmith Functional Motor Scale. It is expanded for the assessment of the SMA, spinal muscular atrophy. It assesses 33 different motor functions, scoring them, each of them on the scale from 0 to 2. And you can see the list of the motor functions on the slide. Another symptom requiring objective evaluation is, of course, epilepsy, for which we suggest using the Early Childhood Epilepsy Severity Scale, ECHES for short, or the Global Assessment of Severity in Epilepsy, GACE for short. Other scales that can assess the child's general motor function include the Gross Motor Function Measure or the Pediatric Evaluation of Disability Inventory. Last but not least, we performed blood sample collection. Since the children had to remain fasting for the blood draw, we made sure to complete this step as quickly as possible. We collected around 3 milliliters of blood for PBMC isolation and further immunology studies, approximately 5 milliliters of whole blood for lipidomics, metabolomics, and proteomics, and 2 milliliters into a PAX gene RNA tube for RNA sequencing. All samples were then frozen at minus 80 degrees Celsius. Full details on the blood collection procedure are included in the study protocol, which I would be happy to share with the interested parties. After the blood collection, we made sure to cheer our patients with some threats. Given the limited size of our study cohort, we cannot draw definitive conclusions about the PURA syndrome from our study. However, our study serves as a pilot for a standardized clinical assessment of PURA syndrome and can be expanded into other areas. We collected general data on 6 of our patients together with their variant codes in order to assess genotype-phenotype correlations. We have also collected photographs of the patients' faces to assess dysmorphic features. This graph summarizes the most prevalent features among our patients. As you can see, hypotonia and intellectual disability was the most prevalent symptoms, while epilepsy was present in only one patient. Some of our patients also presented with heart malformations, feeding difficulties such as gastroesophageal reflux disease, excessive weight loss, excessive drooling, or swallowing difficulties. We have also analyzed the neurodevelopmental milestones, which you can see on the slides. In the blue squares is the typical range or the normal range for the age for the milestone development, and on the red dots symbolize the age when the children develop the particular milestone. And as you can see, most of our patients were more or less late in the milestone development. We have also analyzed the growth charts of 2 available patients, and one of them was below the medium line, and the one was developing normally until the age of 5 years old. We have also analyzed the dysmorphic features, which you can see on the slide. And as you can see, in all of our patients, we observed a myopathic face, a high arched palate, open mouth posture, low set auricles, and tended upper vermilion border. These findings are consistent with the previous descriptions of the PURA syndrome. To summarize, within 3 months, we established a pilot study using an original survey and other tools to assess the medical condition and neurodevelopment of our patients, as well as tools to evaluate hypotonia and monitor their clinical state. We have created a solid foundation on which to build a natural history study of the Polish and other patients cohort. During the follow-up, we plan to use improved tools for hypotonia assessment based on spinal muscular atrophy scales, and to work with a neuropsychologist to conduct a full neurodevelopmental analysis using additional scales. We would like to also invite you for a collaboration if you or your close ones would like to participate in our natural history study. This is Dr. Michał Bielak, who has conducted the study. He's sitting right there. Here's an email. We function at the Medical University of Lublin. And on the right, you can see the email for our foundation. This email, I will answer this email, and then we can arrange the meetings or examination. This is also the QR code for our Fundacja PURA Polska webpage. Thank you for your attention.